Hello, my lovely anatomists and physiologists. Michele Glass here, and thank you for joining me for a video on the cell. So when we talk about human cells, we're of course animals, and all of our cells belong to the category called eukaryotic cells. So actually, all cells on the planet can be divided up into one of two categories. They can either be prokaryotic, which means no nucleus, no membrane-bound nucleus, or they can be eukaryotic, which literally means true nucleus. And so as we're looking at our cells, we're of course expecting to see, that important feature. So we can think about our cell as having our cell membrane. So I'm just drawing like a big circle here to represent my cell membrane. And I'm going to draw in the center very often. This is going to be a darkly stained nucleus. And the nucleus is all important. So important that we name all of the cells on the planet based on whether or not they have a nucleus, because this is going to be our where our DNA is stored. Now, DNA we've introduced before is a nucleic acid. DNA, remember, stands for deoxyribose nucleic acid. We talked about it as being our hereditary molecule. We have another video talking about how the cell uses that DNA molecule in order to build proteins. And so what we can also say about the nucleus is that this is where the process of transcription occurs. And this process of transcription is one of the important parts of how proteins are made, and that's going to show up in its own video, right? So we'll be talking about that in the future. We've also already said that all of the fluid inside the cell can be described as intracellular fluid, which we can abbreviate as ICF. And now we can introduce another name for this fluid called the cytosol. What we'll see is that in addition to the nucleus, the cytosol has what we can call organelles. And these organelles are basically small, usually membrane-bound structures that have their own specialized anatomy and physiology, much like the organs in your body. And so this term organelles is talking like tiny organs, kind of with this idea. When we talk about the cytosol and the organelles, basically everything between the cell membrane and the nucleus, we call all of this collectively cytoplasm. So the cytoplasm is the fluid, is the organelles, everything between the cell membrane and that nuclear envelope. That would be called the cytoplasm. So again, this is our basic breakdown for our cell. As we look at these organelles, our job here is to basically be able to describe their function and have an idea of how they work together. What we can talk about next is what is described as the endomembrane system. The endomembrane system is the collection of organelles that are involved in like producing and packaging and exporting materials. So anything that's being made in the cells being made at one of these organelles, anything that's being packaged up, remember those vesicles, is going to be packaged up here. Anything that's going to be exported is happening, basically with this system here. So we're going to see actually 4 key organelles. So we have the, if I can get a little space there. Okay. We have the endoplasmic reticulum. Sorry for all that. Endoplasmic reticulum, which can be either smooth or rough. We have the Golgi apparatus, and we have lysosomes. So let's talk a little bit more about each of these, and then let's kind of construct a picture of what they might look like in the cell. Goodness, messing up my system today. All right. The key feature of the endoplasmic reticulum is that we're going to see this being continuous with the nuclear envelope. We're going to see it continuous with a nuclear envelope. And we'll see that it consists of both smooth and rough ER. And the difference here is that the rough ER has ribosomes and the smooth ER does not. Now, ribosomes are organelles. They do not have a membrane, and they are found in all cell types. So ribosomes are found in both prokaryotic and eukaryotic cells. And this is actually going to be where proteins are made. The process of translation happens here. And so rough ER is called rough because it contains ribosomes. Ribosomes are a structure we should be able to talk about. Okay, so let's make sure we're paying attention to those ribosomes. Now going back to the rough ER, the rough ER is not making proteins, but it will modify newly made proteins. Remember, we talked about glycoproteins as being proteins with carbohydrates attached. If a carbohydrate is attached to a protein, the protein was made at the ribosome, and then the carbohydrate is attached at the rough ER. So this would be an example. The smooth ER is making a bunch of stuff. So smooth ER is going to be making phospholipids. So it's going to be a key player in the construction of membranes. It's going to be making steroids, such as estrogens and testosterone. So you'd expect to see a lot of smooth ER in gonads, gonad cells. It's going to have this job of sequestering calcium ion. Sequester means store and hide. So we'll see this especially in muscle cells and in neurons. And then it also is going to detoxify drugs and metabolites, which means byproducts of metabolism. So we do see that the liver cells are going to have a huge number of smooth, that grammar was messed up. We'll see the liver has a lot of smooth ER. That's what I wanted to say here. Okay. When we look at the Golgi apparatus, the Golgi apparatus really has the job of sorting, modifying, shipping, things that have been produced at the ER, modified by the Golgi, and then shipped by the Golgi. It's going to have an appearance like stacks of membranes, so we'll show that in our drawing in a few minutes. And it's going to have both a receiving side and a shipping side. So as the molecules move through the Golgi apparatus, they're being changed. And when we talk about shipping, it's going to be making vesicles. So things that are packaged up in vesicles have been processed by the Golgi apparatus. And then the last one here is a lysosome. So remember that prefix lys is talking about breaking, right? We talked about that with tonicity. That ending some means body. So here it's just talking about a vesicle, really. So this is a vesicle of digestive enzymes. So lysosome is a vesicle of digestive enzymes. So these enzymes are really important. If you have like old worn out pieces of organelle that you need to destroy, then a lysosome confused with that basically engulf it and break it down. When we talked about phagocytosis and we said those white blood cells are engulfing your pathogens, the lysosome is how those pathogens are digested. So it's important that the lysosome is packaged up because the digestive enzymes inside can actually break down that cell that made it. So we actually have this term here, autophagy, which is talking about cell digesting its own structures. So again, maybe because, you have a worn out organelle or pieces like that. We have, again, breakdown of pathogens. Again, so we're going to see lysosomes in any kind of phagocytic cell. And then we have this other term here called apoptosis, which is actually one of my favorite science words because it sounds so fun and it's so dark. Apoptosis is described as programmed cell death. So what happens when apoptosis is triggered is that these lysosomes open up, release those digestive enzymes, and digest the cell from the inside. So really grim. Okay, let's see if we can picture. Let's see if we can draw a picture. So let's begin by drawing our outer border to our cell. So just kind of like a big circular cell. Animal cells don't have like a perfect shape, right? So it doesn't have to be like a nice, perfect circle. And then let's represent our nucleus at the center of the cell. And let's just write a note for ourselves that this is holding the DNA molecule. Now, if you want to, the nucleus has a nuclear envelope, which then, let me see. Has little holes in it. I'm gonna try one more time to get my little holes. Read it this way. Yeah. So I'm actually there. My little holes are becoming big holes, but okay, you get the point there. So the nucleus has a nuclear envelope, and the nuclear envelope does have some pores that does allow for small things to move through. Okay. Then right up here associated with the nuclear envelope, you want to draw your endoplasmic reticulum. Endoplasmic reticulum kind of looks like a labyrinth. So just kind of take it back and forth. Looks like it's a little path that goes back and around. And then what we'll see is that the rough endoplasmic reticulum will have ribosomes. So we can describe ribosomes as being attached when they're embedded in part of the rough ER. But some of our ribosomes will actually be free in the cytoplasm or cytosol. So we'll have ribosomes that are attached and ribosomes that are free. Then we can draw our Golgi apparatus. So whereas the endoplasmic reticulum was like kind of a labyrinth, a path you could take, your Golgi apparatus is more like stacks of pita bread or stacks of pockets. And so you can draw these kind of like these small oval shapes stacked together. Remember, you have a receiving end and then a shipping end. So the receiving end is going to be oriented towards the endoplasmic reticulum, and then the shipping end would be oriented toward the cell membrane. And what we can actually see happening here is you might make a protein and package it, modify it, package it at the ER, ship it over to the Golgi, and then the Golgi, so it's going to travel through the layers of the Golgi until you package it up and prepare to ship it out, right? So we looked at exocytosis before, and that would be an example of what would be going on with that. So this is essentially endomembrane system. Maybe that vesicle is actually a lysosome, and so it's ready to digest either broken pieces of the cell itself, pathogens, or perhaps be involved in apoptosis. Okay, the next round of organelles to talk about are involved in energy production. And so here we have an organelle called the mitochondrion, is the singular version of the word. I typically say mitochondria, which is plural. Cells typically have multiple. And then the next one to talk about is the peroxisome. So the mitochondrion is often described as the powerhouse of the cell. This is where ATP production occurs. And the process here of ATP production is called cellular respiration. And really, our book doesn't go into a lot of detail of this process at this point. And what we do need to know is that cellular respiration is going to require oxygen. So in order for a cell to make ATP at the mitochondrion, it's going to need oxygen. So we can also talk about this process as being aerobic, where aerobic means oxygen is required. Now, peroxisome has that some ending, which again means body, but here it has perox at the beginning, as in like hydrogen peroxide, actually. So peroxisomes are going to be vesicles of enzymes, similar to what we saw with the lysosome. But these enzymes are going to be involved in lipid metabolism, meaning breaking down lipids, and chemical detoxification, particularly neutralizing alcohol. So again, you might see a large number of peroxisomes in liver cells for that reason. Basically, what's happening here is you're going to see the transfer of hydrogen ion to oxygen, which basically produces H2O2, which is hydrogen peroxide. And then hydrogen peroxide can be broken down into water and oxygen gas. And so you're taking a potentially hazardous molecule, hydrogen peroxide, and turning it into water and oxygen. So no big deal. Essentially, what we're dealing with here are what are described as reactive oxygen species, or ROS. And another way of talking about these is free radicals. And these are like hydroxide, these are H2O2, and this is what's called superoxide, which is oxygen with a negative sign. And all of these can really disrupt your body chemistry. They can cause what's described as oxidative stress, which is linked with DNA mutations in cancer. So the peroxisome is involved in helping to eliminate these reactive oxygen species that are just a byproduct of metabolism and then converting them into something that's safe for the cell. So let's add to our earlier drawing. Let's draw some mitochondria. So mitochondria are sort of like jelly bean or kidney shaped. They have a double membrane, and then they have a lot of folding. And so you typically get something that's shaped kind of like this here. So these are the mitochondria. Remember, that's going to be where ATP is produced. That's the process of aerobic or cellular respiration. Okay, next we want to talk about the cytoskeleton. So the cytoskeleton is really just composed of fibrous proteins. Remember, we talked about proteins being involved in structure to the cell. We're going to see it involved in just structural support of the cell. We're going to see it involved in cell motility. So this might be actual movements of the cell itself, but this could also be internal movements. Remember, we're going to see proteins being made in the ribosome, modified in the rough ER, packaged. That packaged is moved to the Golgi. It's going to move through the Golgi, and then it's going to go to the cell membrane for export. So there's a ton of internal movement that's happening. Cytoskeleton is involved in cell reproduction. So we're going to see it's involved in moving around chromosomes during mitosis. And then we can also, I guess here the term transportation is talking more about that cell motility, that internal cell motility, in like that endomembrane system. We have 3 different types of fibrous protein filaments. So we have what's called the microfilaments, and here we're going to see actin and myosin, and these are going to be huge in muscle contraction. So we're going to come back and talk about these a lot in the future. Intermediate filaments are going to be more involved in anchoring organelles. So we have these intermediate filaments that are attaching organelles to each other, attaching organelles to cell membrane. It's very much these intermediate filaments that often make these little like pulley sort of pathways between organelles as we talk about like those movements of those vesicles. And then microtubules we'll see on cilia. We'll see in flagella. So here are your cilia. We saw pseudostratified ciliated columnar. So we had the cilia at the top that actually move back and forth. So they can move that way and then that way, right? So they move back and forth. We have flagella on our sperm cells that let them swim. And then we'll also see here the centriole, which is going to be an important track to move chromosomes during mitosis. And as I say this and write this, I think oh my goodness, they're going to be like, what did she say? You know, because we haven't talked about that at all yet. So that's coming up. Mitosis is one of our last videos of chapter 3. So we'll totally get to that point, and chromosomes will be defined then, but basically we'll see the centriole and as such then the microtubule involved in cell division. Okay, that's it for our organelles. So we're not going like super duper detailed. We just need to have a general idea about each of these structures, and stay tuned for protein synthesis. And as always, take care of yourselves and each other.